Good afternoon. Thank you so much, Els, Alice. Thank you so. Hi, folks. Good afternoon. My name is Pedro, and currently I am a master's student in college, but at the National Institute of Amazonian Research in Manaus, Brazil. Today, I will present the results of my master's thesis, which is in class, what is the role of environmental heterogeneity in the evolution of the blue-crowned Manakin. And this research was advised by marine ancients and co-advised by Dr. Igor Keffer. So the phenotype can express several characteristics of the organisms, such as healthy, physical capacity, age, and sex. And individuals and populations can show phenotypic difference. This phenomenon is called phenotypic variation. This can happen within population and between populations. And this variation is the main material to act the natural and sexual selection. With this, the focal species of this research is the blue-crowned Manakin. Belonging the family Pipridae, this species presents, besides the sexual dimorphisms, a great phenotypic variation, in which the plumage of adult males varies from green to black, with many shades of blue in the crown throughout the distribution. This variation has been targeted of research mainly relating this variation to the genetic structure of this group. However, the phenotypic variation doesn't match the genetic structure found. Two studies investigate this, Chevron and collaborators and Hayes and collaborators, but with no relation between coloration and genetic. Based on this, and because of the wide range of environmentals that species occupies, we investigate if environmental factors influence genetic and phenotypic diversity in the blue-crowned Manakin. For this, we built a database with 259 spectral samples in the locations with had Los Angeles on this map, and 343 genetic samples from the locations in yellow Los Angeles on this map, and another 1,200 occurrence data to generate the environmental data. We take an integrative approach to analyze all data. For spectral data, we calculate the JNT using the bird's vision model, build the phylogenetic relationship, and then estimate the phylogeographic relationship. Run the ecological niche models to calculate the environmental data and estimate the last environmental cost corridors. Our analysis generated distance matrix that served as inputs for multiply matrix integration with randomization. So we found 4 well-structured transcendent lineage, South American lineage, North Amazonian lineage, and African lineage. We find with haplotypes network, high cis and transcendent structure between cis and transcendent, and not much structure in the Amazon basin. The ecological niche models show high suitability in the Japra River area, which is considered an environment holds on suitability map of the LCCP. And the MMRR genetic with the genetic distance among locations is explained by the geographic climate and least environmental cost and distance. In other ranges, the variation coloration is not explained by either geographic, climatic, and the least environmental coast distance. And our conclusion is that the genetic diversity is determined by the geographic and climatic distance. This indicates the role of both neutral diversity and adaptation in the evolution of the Blue-crowned Manakin. The color diversity didn't respond to geographical and climatic distance at the third scale. Stochastic and deterministic processes may operate at local scale in the evolution of adult males' plumage color in the Blue-crowned Manakin. And we thank the institutions, the museums, and their curators, and the founding agencies involved on this study. Thank you so much.